Okay, so we're continuing with 17.9. I'm going to finish this chapter out. And we want to talk about the different types of white blood cells. In laboratory, you've been introduced to this, and as a matter of fact, if you've had your first quiz, you've already had to memorize some of this. So white blood cells are also called leukocytes. Now, I notice a lot of people confuse the term leukocyte with lymphocyte. Don't do that. A lymphocyte is a type of leukocyte. Leukocyte is just the general term for white blood cells. Okay. They have nuclei, they have other organelles, unlike the red blood cells, but they don't have hemoglobin because they have a whole different purpose. And there's 5 types, and you can remember these with mnemonic: never let monkeys eat bananas. Or you can make up your own mnemonic: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And the ones we call granulocytes that have granules in it are the neutrophils, eosinophils, and basophils. The ones that are agranulocytes are the monocytes and lymphocytes. So that's one way to recognize at least that difference at the beginning in the microscope as to whether you see granules throughout the cell. Now, realize that white blood cells circulate only for a short portion of their lifespan. The main purpose of using the bloodstream is to travel between the organs and to get to the tissues where you have infection or injury. The white blood cells do spend most of their time migrating through the loose and dense connective tissues. And again, their purpose is to find the infectious agents. All the white blood cells can migrate out of the bloodstream. When they're circulating in the bloodstream, they become activated, and they contact, they adhere to the vessel walls, they squeeze between the adjacent endothelial cells, they go into the surrounding tissues, and this is called immigration or diaphoresis. Diaphoresis is the term that I see used most often. All the white blood cells are attracted to specific chemical stimuli, and we call this positive chemotaxis. That guides the white blood cells to the invading pathogens in the damaged tissues. Now, when we get into the chapter on the lymphatic system, we will talk about these chemotactic factors. Whenever there's damage to tissues or there's infection at certain sites, some of the tissues and some of the other white blood cells will give off these chemicals that automatically signal the white blood cells to come into those areas. The neutrophils, eosinophils, and monocytes are all capable of phagocytosis. What does that mean? What is a phagocyte?
They engulf debris.
They engulf, they actually wrap around and bring it in, right? And they do different things with it, but for the most part, their purpose is going to be to degrade eventually. Although monocytes, and we'll talk more about those in that chapter on the lymphatic system, the monocytes will turn into macrophages, and they have other uses like displaying the antigen from that organism to T and B cells later down for a specific immunity. So the macrophages are the monocytes that have matured to that level, and they move from the bloodstream into the peripheral tissues. The number of white blood cells tells us a lot about what's going on. The numbers increase when you have infection or inflammation or an allergic response. And we also can tell a lot by the type of white blood cells that we see. So this is a white blood cell differential count, and we report it as a percentage per 100 white blood cells. Okay, again, we have 2 classes of white blood cells, either the granulocytes or agranulocytes. The granulocytes have lots of cytoplasmic granules, and what these really are secretory vesicles and lysosomes. You might remember from 168 that they are different types of vesicles that either will have some kind of digestive enzyme in them, or in the case of secretory vesicles, their purpose is going to be to release something out of the membrane. And they do absorb certain histological stains, so they do look different in the microscope when we have certain stains that we're using. The agranulocytes tend to be smaller. Again, The secretory vesicles and lysosomes here don't absorb the stains, so you don't see those granules. So let's look at each of these. Here's your monocytes. They're really large cells. The nucleus is like a large kidney bean, and they fill up a lot of the cell. They enter the tissues and become macrophages, where they will engulf pathogens or debris. They move into the tissues after 1 to 2 days. They survive for months or longer, and these are produced mostly in the red bone marrow. Okay, the lymphocytes. They're generally round cells. They're slightly larger than red blood cells, and you can observe them by their very round nuclei. They're not going to have the many different shapes or morphologies when it comes to the nucleus. It's usually one of the more obvious things about it. They are cells of the lymphatic system. They provide defense against specific pathogens or toxins. So one thing you want to remember about lymphocytes is they're involved in specific immunity. And what that means is they actually recognize very specifically particular antigens, where the others are much more general. They're just going to go after anything that shouldn't be there. The lymphocytes are very specific. They target things. They survive for months to decades, and they circulate from the blood to the tissues and back. They're produced in the red bone marrow and the lymphoid tissues. Did I miss some? No, okay. Okay, let's talk about the Clyde response. Actually, I do want to go over the rest of those white blood cells on page 647. I guess we're just missing some of those from this PowerPoint slide. Okay. Let's look at the page, which is page 647. I want to go over the rest of them. So the two that we already mentioned in this slide are the monocytes and lymphocytes, and they are both agranulocytes. The granulocytes, on the other hand, yeah, I'm sorry the slide's not up here. The neutrophils, they're round cell. The nuclei are lobed, and one way to recognize them and differentiate them is that the nuclei resemble a string of beads. The cytoplasm also contains large pale inclusions. They are phagocytic. They engulf pathogens or debris in injured or infected tissues. And one of their purposes is to release cytotoxic enzymes and other chemicals. They move into the tissues after several hours. They can survive for minutes to days. They're produced in the red bone marrow. The eosinophils. They're a round cell. In this case, you will recognize them by two lobes. So we say they're bilobed. They have large granules. They generally stain a bright red color. Again, they're phagocytic. They engulf antibody-labeled material. They release cytotoxic enzymes. They are involved in inflammation, and they are often found in larger numbers when you have allergies or you have certain parasitic infections. So that's a way to recognize that. They move into tissues after several hours. They can survive for minutes to days, and they're produced in the red bone marrow. And then the basophils, they're round cells. The nucleus is generally, you cannot see it because they have so many granules. They enter the damaged tissue, release histamines and other chemicals that promote inflammation. And the survival time is unknown in this case. They assist the mast cells in producing inflammation. So they actually help in the inflammatory response. They don't reduce inflammation as the eosinophils do. And they're produced in the red bone marrow. Okay, now we'll move forward on the slides here. Hemostasis. This is a process responsible for stopping blood loss through the walls of the damaged blood vessels, and it establishes a framework for tissue repair. This is very important, and I mentioned this when we first started talking about blood. There are 3 ways you prevent blood loss. If I asked you, or if I had asked you, you probably would have come up with the first one, which was simply what? Clotting. You'd probably say blood clots. Well, there's actually 3 ways that your body can prevent blood fluid loss. Okay, and certainly the use of platelets is one of the ways, but there's also a coagulation phase, and there's a vascular phase, and we're going to go over each of these. Okay, let's start with the vascular phase. It lasts for about 30 minutes after the injury, and it is an endothelial response. It involves a vascular spasm where the smooth muscle contracts. So all you're doing is contracting the vessel so that not as much blood goes out of the vessel. It's like squeezing the hose pipe, but really tight so it can't go out. Like kinking the hose pipe, I guess. The exposed endothelium is in contact with the blood. You release chemicals and local hormones. So that's the vascular phase. Continuing with that, the endothelial cells then release chemicals and local hormones. They include the endothelins. This is what causes the smooth muscle contraction and promotes vascular spasms. It also stimulates the division of endothelial cells and smooth muscle cells and fibroblasts for the repair mechanism. And this also causes the endothelial plasma membranes to become sticky, and that's important in the repair process. We'll talk more about repair processes in another chapter when we talk about inflammatory response. Okay, platelet phase begins with the attachment of the platelets to the sticky endothelial surfaces, the basement membrane, the exposed collagen fibers, and other platelets. The platelets themselves release certain chemicals. Adenosine diphosphate stimulates the platelet aggregation and secretion, and these chemicals stimulate vascular spasms. We have platelet factors that are released. These are proteins that will be involved in the clotting process. We have platelet-derived growth factor that promotes vessel repair. Calcium ions are also involved. These are required for platelet aggregation clotting processes. Okay, and then we have the coagulation phase. It starts 30 seconds or more after damage. So coagulation is blood clotting. It involves a complex sequence of steps that lead to conversion of the fibrinogen that circulates in the blood to an insoluble fibrin. And what then happens is that the blood cells and the platelets get trapped in this network of fibrin, and that's what forms the blood clot. Procoagulants, which are clotting factors, also play a very important role. There's calcium involved with 11 different proteins. And number 1 through 11 is the way we label them. A lot of them are proenzymes, which are inactive, but they become activated. And they cause a chain reaction or a cascade. There are 2 pathways that lead to a common pathway, or the extrinsic and intrinsic. The extrinsic pathway begins with the release of tissue factor, which we call factor 3, from the damaged endothelial cells of the peripheral tissues. Then the tissue factor combines with calcium and other clotting factors to activate factor 10. That's the first step in the common pathway. The intrinsic pathway begins with activation of proenzymes exposed to the collagen fibers at the injury site. And then the pathway continues with PF3. This factor is released by the aggregating platelets. And then the sequence of enzyme activations leads to factor 10. Okay, the common pathway. The activated factor 10 activates prothrombin activator, and that's a complex that converts prothrombin to an enzyme that's called thrombin. Thrombin converts fibrinogen to fibrin, and that completes the clotting process. Now, you have to have clot retraction also. The red blood cells and the platelets stick to the fibrin threads. The platelets will contract to form tighter clot and pull the edges together, and that continues for 30 to 60 minutes. So this shows you that entire process. The extrinsic pathway on the left, the intrinsic pathway on the right, and then the common pathway in the middle. You'll notice the differences are at the very beginning in what factors are used and how they get started. But they both lead to factor 10. And then all of them go to the prothrombin activator, where the prothrombin is converted to thrombin, and then you have fibrinogen converted to fibrin eventually. So again, the difference is where it begins and the types of factors that are involved. Now eventually, and you don't want clots to last forever, you have fibrinolysis. So that's the clot dissolving. And it begins with an activation of plasminogen by thrombin. The tissue plasminogen activator, also called TPA, comes from the damaged tissues. It produces plasmin, which erodes the clot. Okay, a lot of different chemicals involved there. There's kind of no easy way around it but to remember that diagram on page 649. But as usual, look at what I've asked you to remember in the study guide for the exam to get what details I expect out of you for that. Okay, so we can tell a heck of a lot about what's going on in a person by their blood. And one way you can obtain blood for diagnosis is basically the venipuncture, withdrawal of whole blood from the superficial vein, like the median cubital vein. And you'll learn these in the next chapter. I'll be giving you a whole list of arteries and veins to know, kind of like you had to know the bones and muscles and various. So we give you specific ones you have to remember. The venipuncture is commonly used because it's easy to locate the superficial veins. Vein walls are thinner than the walls of the comparable arteries. Venous blood pressure is relatively low, so the vein seals itself pretty quickly. You really don't want to hit an artery if you don't need to. And you know it if an artery's been hit, because it is under pressure. I don't know, have you all heard in the news lately, this lady who's kind of a self-made billionaire, I think actually, had created this system of blood tests that could recognize, I don't know, 50 to 60 different disorders simply by this really quick little test. And I forget the name of this company. It was on the news last night. Oh, can't remember. But they found out that a lot of her claims were false, and now there's a big suit against her. Has anyone heard of this? But it showed so much promise. She had a lot of big backers for this particular test because it was so simple and it would tell so many things supposedly. But the problem is it started telling people, giving people false positives on a lot of different issues. So we have to be careful about, obviously doing blood testing for many things. Now, one thing that we can tell, iron deficiency anemia. If you have insufficient iron intake, you need that for the functional hemoglobin so you can attach to oxygen and carry that through your system. And what happens in this case is that your red blood cells are small, or what we call microcytic, so you can't transport as much oxygen. This tends to be more common in women. Iron reserves are half the reserves of a typical man. Then there's pernicious anemia. In this case, vitamin B12 deficiency. prevents your normal stem cells from dividing. Fewer red blood cells are produced, and then also when you're misshaped and large, and then they're called macrocytic. This can be from the lack of intrinsic factor that's secreted in the stomach, and that's needed for vitamin B12 absorption. So maybe you're getting enough B12 in your nutrition, it's just that you're not absorbing it well. And then calcium and vitamin K deficiencies. Calcium is required for all the clotting pathways. Vitamin K is required by the liver to synthesize the clotting factors. Sickle cell disease. These are inherited red blood cell disorders caused by genetic mutation that affects amino acid sequence of the beta globin subunit and the hemoglobin. And we call it sickle cell because it does cause the cell to take on this funny shape when they release their oxygen. It causes the red blood cells to be rather fragile and easily damaged. It also causes the red blood cells to get stuck in the smaller vessels, and you can get a block in the flow of blood through the vessels. Sickle cell anemia is the more severe of sickle cell disease. It requires 2 copies of the sickling gene. If you have just 1 copy, you have the sickling trait, but you don't have the full disorder. But having the trait still increases the person's resistance to malaria, and this is why it still stays predominant in certain areas of the world, or it stays around in the population, because it does confer this particular resistance. And what happens is the infected red blood cells will sickle, the sickle cells will be destroyed by the macrophages, and of course along with that the malarial pathogen will be destroyed. So that's why it's actually a benefit to at least have one of the genes in certain parts of the world. Hemophilia. This is an inherited bleeding disorder. Affects 1 person in 10,000. 80 to 90% of the people that have it are males. It's caused by missing or reduced production of a clotting factor, and the severity is different from person to person or family to family. In the severe cases, extensive bleeding occurs with a minor contact. Bleeding also occurs at joints and around muscles. One of the famous families, the royal families that had it, the Habsburgs in Europe, they often didn't live very long. One reason why that trait stayed around, because it was a recessive disorder, is because the royal families tended to keep the genes in the family, so a lot of inbreeding in some of those families. And what would happen often is a simple cut would cause one of them to bleed to death. So, like I said, they didn't live very long. Thalassemias, this is a diverse group of inherited disorders. You're unable to adequately produce normal hemoglobin protein subunits, but it depends on how many of the subunits are abnormal in the person as to whether it causes a problem or not. And of course, we can tell a lot from blood as far as infections. Bacteremia is when you see bacteria circulating in the blood, and viremia when you see viruses circulating in the blood. In both of these cases, the organisms are not multiplied in the blood, though. Okay, sepsis you have heard of. This is when you have widespread infection of the body tissues. Septicemia is sepsis of the blood, or blood poisoning. In this case, the pathogen is present and you have multiplication of the organism in the blood and it spreads. In this case, you see an individual with meningococcal bacteria, one that causes meningitis. Okay, so we had mentioned that sickle cell having one of the genes would actually help you. And malaria is a parasitic disease caused by several species of Plasmodium, which is a protozoan organism. It's a one-celled organism with, I'm going to forget here, it's either a ciliate or it's just or it doesn't have motility. I'm forgetting all of a sudden. But in any case, it is a type of protozoan that's under the kingdom Protista. It kills 1.5 to 3 million people per year. Up to half are under age 5. Transmitted by mosquito. Initially infects liver, later infects red blood cells. And every 2 to 3 days, all the infected red blood cells do rupture, releasing more of the parasites. And this is what causes a cycle of the fever and chills. You know, every time you have this rupturing of the cells, the dead red blood cells block vessels to the vital organs. So it is very serious, or can be. In the early years of exploration of the United States, especially here in the southeast, it was not uncommon. Leukemias, cancers of blood-forming tissues. Cancerous cells spread from origin in the red bone marrow. The first symptoms appear with the presence of immature and abnormal white blood cells in circulation. It's fatal if it's untreated. And there's 2 types, myeloid and lymphoid leukemias. In both cases, you have elevated white blood cells. Degenerative blood disorders, disseminated intravascular coagulation. Here we have bacterial toxins that activate certain steps in coagulation, which then converts too much of your fibrinogen to fibrin, so you have small clots that block your small vessels. And phagocytes and plasmin work to remove the fibrin. The liver is going to try to maintain the adequate fibrinogen levels, but if it can't keep up, you then have uncontrolled bleeding. And that's it for this chapter.